In this lesson, we are going to study prokaryotes, which are organisms that have cells with no nucleus or other membrane-bound organelles. There are two kingdoms that are completely prokaryotic. The first kingdom we'll talk about in this slide is kingdom Eubacteria. They are all unicellular, meaning that each organism is only comprised of one cell. This makes them microorganisms. Bacteria are very small, typically in the range of 0.5 to 5 micrometers. Their cellular structure is, of course, different than the cellular structure that we have. They're, as I mentioned, prokaryotic with no membrane-bound organelles, so they would have no nucleus, no mitochondria, and no chloroplasts. And you can see this in the diagram on the slide. You would see there is DNA in the middle, but no nuclear membrane that surrounds that DNA. Speaking of their DNA, it is also organized into a single circular chromosome. This is different from us as well because we have several different chromosomes, and it's not arranged in a circle. It's just linear. Their cell wall is also unique. Bacterial cell walls are made of a complex carbohydrate called peptidoglycan. And this helps us to separate bacteria into two different groups based on how they react to a procedure called Gram's staining. A gram-positive bacteria possesses a thick cell wall containing many layers of this peptidoglycan. Gram-negative bacteria have a relatively thin cell wall consisting of a few layers of peptidoglycan surrounded by a second lipid bilayer membrane. Bacteria have several different morphologies or shapes. The three main shapes that are usually associated with bacteria include coccus, bacillus, and spirillum. The coccus bacteria are sphere-shaped, as you see some examples of this in the lower left-hand corner of the slide. Bacillus are rod-shaped bacteria, also found on the bottom of the slide but towards the middle. And then we have spirillum, which would be spiral-shaped. Those are found in the lower right-hand corner of the slide. Now, bacteria can also have different arrangements that also go into making up their shape or morphology. For example, you have diploids, which are bacteria arranged in pairs. Strepto, which would be arranged in chains, and Staphylo, which are clusters, and typically are found in the cocci bacteria that are arranged as like grapes. Some of these names may be somewhat familiar to you. For example, if you know that you have an infection of strep throat, that's because you have streptococcus bacteria that is infecting your throat and making it extremely sore and making you sick. That is a long chain of round bacteria that causes streptococcus. There's also staphylococcus, which gives us staph infections, which can be pretty dangerous. So these are all different morphologies or shapes of bacteria. Bacteria also have various forms of metabolism. This is, of course, different than us because we are strictly heterotrophic, being that we consume our food. Now, bacteria can be heterotrophic, as you can see in this slide. They would consume their organic carbon sources. But they could also be photoautotrophs. Some bacteria do this by fixing carbon dioxide into organic carbon that they use for food. And they would use photosynthesis for doing this. Some bacteria are also chemoautotrophs, where they do the same thing. They fix carbon dioxide into organic carbon, but rather they use chemical energy or chemosynthesis to do this. Bacteria reproduction is much more simple than the reproduction we see in other forms of life. Bacteria primarily rely on binary fission, which is a form of asexual reproduction, and is really a very simple process where the DNA inside the cell copies itself, and then the cell splits into two. It's different from mitosis in that there's no nucleus to divide. It's just one chromosome of DNA that it copies itself, and then the cell splits, taking a copy of each of those chromosomes with it. You may wonder, well, if it's strictly asexual reproduction, how do bacteria mutate? How do they change their genetic identity? Well, this is because bacteria are able to do what's called horizontal gene transfer. So bacteria can actually incorporate the genetic material from another organism into their own DNA. This is different than what we do. We get our genetic variability from our parents because of vertical gene transfer. We inherit genes from our parents, and that makes us unique individuals. Bacteria do it horizontally by simply absorbing or incorporating genetic material from, say, another bacterium or something else. Bacteria can be found in just about every habitat on Earth, including soil, acidic hot springs, radioactive waste, water, even deep inside the Earth's crust. We even find it in organic matter, and they quite often live on the bodies of plants and animals. In fact, we have several different species of bacteria that live on us. They live on our skin, and they're called commensal bacteria. And they're perfectly harmless, and in fact, sometimes they help us to ward off infections from bad bacteria. We also have bacteria that live inside our intestines that help us to digest food. In the picture on the screen, you can see some of the unusual habitats where we can find bacteria living. Picture A shows you a microbial mat on a mud volcano. And picture B, this is bacteria in the Black Sea, which is extremely salty. There's also microbes in picture C growing on oil sediments. And letter D is neat. It's an actual microbial mat of bacteria growing on an asphalt flow. So bacteria can grow in any number of places, and they're one of the most abundant organisms on. The second kingdom of prokaryotes would include kingdom Archaea, and that's what we're going to start discussing in this slide. They are also unicellular prokaryotes like bacteria. In fact, they resemble gram-positive bacteria, but they differ from bacteria in a number of ways. Most notably, they have a separate evolutionary history. They're simply a different organism than bacteria. And they were first classified into a separate group in 1977, so it took some time for microbiologists to be able to realize that we're looking at a different life form here. And this was done based on the studies of the archaeans' ribosomal RNA sequences. Another difference with the Archaea is that their origin appears to be very old. In fact, it's thought that the Archaeans may likely represent ancestors of the first, or descendants, of the first organisms that ever appeared on Earth. Thirdly, their cell membrane structure is different from both bacteria and eukaryotes. This difference lies in the phospholipid that goes into the phospholipid bilayer that makes up the cell membrane. Their phospholipid has a different structure than what we would find in our cell membranes and in a bacteria cell membrane. And fourthly, in contrast to bacteria, most archaea lack that peptidoglycan compound in their cell. Now you may think that because archaeans are prokaryotes like bacteria, that they would share a closer ancestry or lineage with one another, but that's actually not the case. Archaeans actually possess genes and metabolic pathways that are more closely related to eukaryotes. By looking at this phylogenetic tree, you can see that Archaea and Eukaryota, or Eukarya as it's called now, are on the same branch of this phylogenetic tree, showing that they have more similarities than they do to the bacteria. Archaeans have a number of habitats, such as soils, oceans, marshlands, and even in the guts of termites. In fact, they are responsible for allowing termites to digest wood. Archaea are particularly numerous in the ocean, and they actually take up an abundant population in the plankton. And as a result of this, Archaeans may be one of the most diverse organisms and most abundant organisms on the planet. Now, some Archaeans are famous for being extremophiles, meaning they can live in harsh environments, such as in hot springs and salt lakes. And of course, as a result of intense study over the past few decades, we finally realized that Archaea are now a major part of life on Earth, which is interesting because we never knew of their existence until the 1970s.